is David Delaney from Cramer Fish Sciences. So, David.
Thanks, Sam. Good morning. You can put the presentation. Great, thank you. Good morning. Today I'm going to be discussing results from the Stipulation Study. The Stipulation Study was conducted because on January 12th, 2012, the plaintiff intervener, and the federal defendants, the consolidated Salmonic cases, signed and filed a stipulation that the acoustic telemetry and certain operations of CVP and SWP would be conducted in April and May of 2012. I'm going to illustrate some of the results from this. There we go. All right, so today I'm going to be talking about some of the findings of the Stipulation Study report, which you see here on the right. I'm only going to cover some of those, so if you want the full analysis, it's been provided to the panel, and it can be downloaded from the website at the bottom right of this slide. So to start off, I'm going to talk about who I work for, my qualifications to analyze this data, and then give you an overview of the Stipulation Study, how the methods we collected data on the movement of acoustically tagged juvenile steelhead, and then examine if the Delta Simulation Model 2's hydroparticle tracking model can predict the migration of these acoustically tagged steelhead. Then I'm going to examine the routing of these steelhead in the Delta to see if they're affected by OMR flows observed during the study. And then I'm going to examine if there's a zone of influence of the export facilities. To conclude, I'll go over all the results, and then talk about some future studies that we feel would be very beneficial to be done in the future. So I work as a fisheries biologist for Cramer Fish Sciences. Our overarching goal is to apply science to understand and hopefully resolve complex management issues such as what's occurring in the Delta. This group was founded in 1987, and they have offices throughout the West Coast. So they've been conducting research for over 25 years. I've only been with the group for a year. I started in 2013. Previously, I was the director of research of an international research center in Africa. I received my PhD from McGill University's Department of Biology, and during my short career so far, I've worked with both active and telemetry studies, which gave me some experience to handle this data set. And this study was conducted from the start of April to the end of May in 2012. And it was a collaborative effort. It involved California Departments of Water Resources, NOAA, NIMS, and consulting groups such as Hansen Environmental, among many others. We released hatchery-raised acoustically tagged steelhead. They were provided from the McCullough River Fish Hatchery. They were released at the Green Star, which is Buckley Cove, which is just upstream of Turner Cut and the San Joaquin River. And this allowed us to collect data on fish movements in the central and south delta. These acoustically tagged steelhead were implanted with Vemco V6 transmitters, which you see in the center of that top right figure. They're 6 millimeters in diameter, 16.5 millimeters long, and weigh approximately 1 gram. This led to a tag burner, the ratio between tag weight and the steelhead, of 0.9% on average, and all fish were less than 2% that were used in our study. We collected data on their movements in the receiver arrays you see here in the bottom right. The red squares are the acoustic receiver arrays that were deployed for the Stipulation Study, and the blue squares are the receiver arrays that were deployed for the Steelhead 6-year study. And we used some of those because those are compatible with our V6 transmitters. We released these steelhead in the following manner. The first release started on April 15th, 2012. We released at least 166 steelhead over 24 hours. That was every 3 hours we released a subset. Then 2 weeks later, we released another subset of at least 166, and then a third set in mid-May. The experimental design was to have certain OMR conditions for each of these releases. The target flows were negative 3,500 cubic feet per second for release group 1, negative 1,250 CFS for release group 2, and negative 5,000 CFS for release group 3, But what was observed as the average flows in the first week of those releases was actually negative 2,446, negative 2,933, and negative 5,038 CFS. Therefore, based on this and the recommendation of the 2012 Independent Review Panel's report, we actually grouped the data from release groups 1 and 2 and considered these less negative OMR conditions and compared those to the more negative OMR conditions examined during release group 3. So when I talk about comparative analysis, this is what I'll be doing, more versus less negative OMR conditions that were observed in our study. So this is how we collect data on fish movements. We want to examine how well they were predicted by the Delta Simulation Model 2's particle tracking model. And before I cover this, I just want to show what it's been used for in the past. Let me just go back a slide. It's been used for a variety of both biological and physical applications. It's been used to examine the effects of various barriers, including the head of old river barrier, which you see in the top right, which was in place during our field study. It's been used to estimate entrainment into various structures, including agricultural diversions and export facilities. It's also been used to make conservation and management decisions, such as done in the 2000 NIMS OCAP biological opinion, restricting OMR flows during the spring. Another use of this model has been to estimate the migration of juvenile anadromous fish in the delta. For example, it's been used for delta smelt and even more mobile organisms such as Chinook salmon. And it's been actually suggested that it could also possibly predict steelhead, which are even more mobile. And like all fish, they have complex sets of behaviors, can swim. So we're not exactly sure this is appropriate, so we tested the null hypothesis you see here, that the mean distance traveled by steelhead tags was not significantly different than the distance traveled by these neutrally buoyant particles as predicted by the DSM-2's particle tracking model. The way we're going to examine this is we're going to compare their distance traveled 3 and 7 days after their release. For example, if you look at these figures, you have some data on steelhead and particles 3 days after the release. For each dot is an array that detected steelhead, tags on the left, and particles on the right. The size of that dot is scaled to the relative proportion being detected at site. And for every particle and steelhead tag detected on that day, we quantified its distance that it traveled by the Euclidean distance from the array that it was last detected at on that day of interest, either day 3 or day 7, to its release site. We analyzed this data, now that we had distance traveled by particles and steelhead, by a t-test for each of the days of interest, day 3 and day 7, separately. So we analyzed it with 2 t-tests in total. Now that we know how we examine the data, let's look at the results. For day 3, we found that steelhead tags were transported significantly further than the neutrally buoyant particles, because particles on average are transported 6.8 kilometers on average, where steelhead tags are transported 9.5 kilometers on average. We found a very similar result for day 7, where again, particles only traveled approximately 70% of the distance of steelhead tags, and the t-test again found this to be a significant difference. Not only is this purely physical delta simulation model particle tracking model underestimating migration rate, it was inaccurately predicting where these steelhead tags were actually being transported to. As we see when we look at the day 7 data, what you see is the black bars are the percentages of particles being detected at the arrays on the x-axis. The gray bars are the percentage of steelhead tags being detected at those arrays on the x-axis. The arrays on the x-axis were sorted based on Euclidean distance away from the release site. So array 1, which is just downstream of the release site, was at the far left. Array 24, which is at Chips Island, is at the very far right. That's our last array before leaving our study system on the way to the ocean. And what we find is that particles are much more aggregated than steelhead tags. For example, 91% of the particles are found at these 6 arrays. And at more than a quarter of the arrays, no particles were detected on that day of interest, day 7. In contrast, steelhead tags were detected at 95% of those arrays, and at 90% of those arrays, they were detected in single-digit percentages. So steelhead tags are much more equally and widely distributed through the system than would be predicted or expected if DSM-2 particle tracking was an index of their movement. And this probably occurs because the steelhead, like all fish, can swim and have complex sets of behaviors that we need to identify and quantify and incorporate into coupled biophysical models that are better able to estimate migration rate and where anadromous fish are migrating to. Not only do we need to build these models, we'll start later. Not only do we need to build these coupled biophysical models, we need to validate them in actual data sets, such as done in the Stipulation Study or the 6-year Steelhead study, or for Chinook salmon, we could use the years from the VAMP studies to really make sure that these models are predicting what we think they're predicting. And then we can use them to make informed conservation, management, and policy decisions. All right, so that was movement of steelhead tags on a large scale. Let's examine their movement at junctions in relation to OMR flows. And we expected that their routing would be related to OMR. When there's more pumping and there's more flows going down to the export facilities, we expected some more steelhead tags to be moving in that direction. So we tested this at 4 different junctions in the delta. This was Turner Cut, Columbia Cut, where the Middle River meets the San Joaquin River, and down in the South Delta at Railroad Cut, which I'll show you in a minute. We use all tags that actually use this junction as we find it, which was being detected at the upstream receiver, which will always be shown as the red dot in these figures, and then being detected at least one of the downstream receivers before leaving the junction, which is delineated by that green circle. And we used the binary response of our generalized linear model as which array of the downstream ones actually detected it last, so its final routing. A significant result would mean that there's significantly more tags routing in a certain direction during more or less negative flows. And if we did find a significant result, we look at those proportions to see if significantly more were actually moving south during more negative OMR conditions over these groups 3. A non-significant result would be no significant difference in the routing between more and less negative OMR conditions that occurred during our study. And now let's examine the results. At Turner Cut, we found a p-value of 0.32, so a non-significant result, meaning that significant tags were not moving south during more or less negative OMR conditions examined in our study. And we found a similar result at Columbia Cut with a p-value of 0.7. Again, no difference between more and less negative flows examined in our study. And at the third junction along the San Joaquin River, again, a non-significant result of a p-value of 0.88. So along the San Joaquin River, we're finding no influence that the OMR conditions observed during our study is affecting routing. But if you look down the South Delta, at Railroad Cut, we find a marginally significant result of 0.08. And if you look at the proportions, you do find more tags actually be detecting at southern array and the more negative conditions found during release group 3 than during the less negative conditions of release group 1 and 2. So there seems to be some marginal, at least significant evidence that maybe there's effective OMR in the export facilities down in the South Delta. If this is occurring, maybe it's because there's a zone of influence of those export facilities shown as these red triangles. And if there is this zone of influence, we need to understand its size. Is there any predictors that predict its size? For example, maybe the extent of the zone of influence is smaller when there's less negative OMR conditions and more northerly during more negative OMR conditions. And we can examine this because we release steelhead during more and less negative OMR conditions. If the zone of influence is related to OMR, we'd expect a relationship such as this, where the zone of influence is more southerly and limited during less negative OMR conditions occurring during release groups 1 and 2, and a larger zone of influence during release group 3 where we had more negative OMR conditions. To examine this in our data sets, we need to figure a way to quantify what is the zone of influence. So we defined it as the area that no steelhead tags were detected, being detected at that array, and then reaching Chips Island without going through SWP or CVP salvage operations. Or alternatively, you can think of this as a line of no return. No tags are detected south of this line, being detected at Chips Island in the end without assistance. And again, if it's related to OMR, within the flows we examine in our study, we'd expect to be more southerly, northern boundary for less negative OMR conditions, and more northerly for more negative OMR conditions. And what we found was the following. We found actually the opposite. During the less negative OMR flow conditions of release groups 1 and 2, we found it slightly more northerly. And during the more negative OMR conditions of release group 3, we found it slightly more southerly. But there wasn't much difference between the two, but there was this area of zone of influence in both figures down the South Delta. So to conclude what we found from the following analysis...
David, can I ask a quick question?
Sure.
How far apart in time were the two releases?
Release groups 1 started on April 15th and ended, and the end of this release group 2 The data collection stopped for that one on May 15th of 2000.
Do you know where they fall relative to springs and neeps?
You have some during that period, both. And so we actually really thought about looking at spring versus neap tides and the effect. But because they occurred throughout the period, we couldn't tease it apart. But it's an important consideration for the future in designing experiments. So what we found was the purely physical particle tracking model of the Delta Simulation Model 2 was not able to accurately predict the migration rate and destination of steelhead tags in our study. This probably occurs because steelhead is all fish, can swim, have complex sets of behaviors. And we quantified some of those and identified some of those behaviors in the stipulation report, but there's others out there. And we need to not only identify those, we need to quantify them and incorporate them into coupled biophysical models to better estimate the migration of juvenile anadromous fish species in the delta. And we also need to validate these on data sets. That's a critically important step that's not done as much as it should be, because that's to make sure that we are actually predicting what we think we're predicting. And then we can use it for conservation and management decisions. We found no evidence that OMAR flows in this study were affecting the routing of steelhead tags in the Sinakim River, but we did find some marginally significant evidence that OMAR flows were affecting the routing in the South Delta. And there was some evidence of a localized and limited zone of influence in the South Delta from our analysis. Okay, so there's a lot of work that still needs to be done to answer some of these management questions in the Delta. So I'm going to talk about two of the ones we talk about in the stipulation report. There's others in that report, but I'm not going to go into today. We need to do some different types of research. We need to actually use a wide range of OMAR conditions to really fully say that OMAR is not playing a role. We had a nice range here, but we could even do larger ones. And we can use larger sample sizes to detect significant differences in all types of studies if they actually exist. We need to give these studies time to be designed in a deliberate and methodical and not rushed fashion. This study was actually put together in a few months, and I think you should give a study like this at least 6 months or longer to design. Also, we have acoustic telemetry data, and some of this data was actually free-swimming smolts, but some of it might have been data from the movement of predators that ate that free-swimming smolt. And for acoustic telemetry studies in the future, we need to be using predator detection tags. HTI just developed some, and the Bureau of Reclamation and Department of Water Resources of California are currently testing those to see how accurate they are in telling the data user when they were consumed by predators. Because for predatory fish like for steelhead, predator filters can cause their own challenges and bias and their own things, so we're hoping these tags will be validated and used in the future. Also, we recommend a meta-analysis approach is done. The delta is extremely well studied. There's decades of coded wire tag data. There's starting to be a wealth of acoustic telemetry equipment, and there's a variety of other experiments being done there. We could actually pool these data sets, look for robust relationships, figure out what we know, and make more informed management decisions based on that. Also figure out what we don't know and what we need to prioritize for future research. Because we need to do this, we took a first step with our own data set. On this website you see here, we've put all the detection histories of all the fish in a web-based tool where you can go there and view the information. The detection history will be shown as this red dot that will move through the system as it pings in through the acoustic receiver arrays. Up in the top right, you're going to get some static information like what's its fish ID number, what release group was it released for, what was its date and time that it was released, and was it detected either at the export facilities and or Chips Island. Down here, I'm going to show you a video of this quickly in a minute. This is going to be dynamic information, like what array is it detected at? What was its day and time when it's first and last detected at that array? How many detections were detected at that array continuously before detected at the next array? And what's its residency time, defined as the time between the last detection at that array and the first detection without being detected at any other array? And how long was it actually detected there after its release is shown here. We have a variety of sorting options, which is the last slide I'm going to show is the next one, where we actually show a little video of this. We had a little technical difficulty, so I'm going to have Robert play an animation to finish my presentation today. And we really feel we need to give the user as much ability to look into this data set to figure out what they want to know. For example, they might want to check the individual tag in our study. You can just press... Great, thank you. So we have 447 detection histories. You could view all of them, but that would take way too long. So we're going to choose a tag from release group 3 because we have 145 detections for release group 3. So we're going to go in here, we're going to pull down a certain tag just so I can show you what a detection history looks like. And we're going to press play, as you see where the mouse is right now. And you're going to start to see that detection history move in the center. Up in the top right, you're going to see that you're going to see that static information. Down here, you're going to see dynamic information, like what array is it at right now where that red dot is? How long was it there, residency time, and so forth. This one ended up at Chips Island. So, one history is not that meaningful. But maybe you want to look at all the detections that occurred at CVP. And that's what I'm going to show you here now. Or alternatively, we could look at all the detections that were detected at SWP and CVP. And you see that's 34 detection histories. And we're going to play all of them. I'm not going to go through all of them, but just to show you that it's possible. And you're going to see the detection histories ping in here. And as they were detected at different arrays, you're going to see static information up here on the top part here, like its fish ID number. This is 11336669. You're going to see dynamic information, so it's down here. And we know this tag is going to be detected at CVP because we sorted the data for detections at that array. After it's done, a new tag is going to ping in the top right. This is the detection history for 11336683, and you're seeing its detection history. We've put a lot of things that a user can do, like they may not like our base map. So you can actually use a satellite map if that's easier for you to make sense of the data. Or alternatively, you might want a road map because you want to know where those southern arrays are and how they're relating to Highway 4. And that's also an option. And you're going to see that there's a lot of things that users can choose, such as the speed at which these tags ping through. You can go step by step to see information on each array. You can make the map smaller. You can create hybrid maps and so forth. So there's a lot of information you can do here. And we really hope that it will actually facilitate future collaboration and getting as much out of these data sets and comparing data sets as possible. So with that, you can move to my last acknowledgement slide. Because there's a lot of people we want to thank. We came here at the end to analyze the data, but there was various agencies and consulting groups that were critically important in carrying out this study in such a short time frame. There were 45 people out in the field. Some of them put in over 100 hours. And some of them put actually 100 hours of overtime in April alone. And they were the ones that actually collected this data and gave us the opportunity to examine this data set. So I just want to take my last slide to say thank you to all these groups who really appreciate all the hard work. And with that, if I have any more time, I'd be more than happy to answer any of the panel's questions. Thank you.
Got about 2 minutes. Quick question.
I have a question about your logistic regression work that you did to see if there's if OMR is affecting the route chain, the route selection. What's the individual sampling unit? What's the response variable there? It's a single fish choosing to go one way or another?
What we did for every fish that actually used that junction, we defined it with the binary. Was it detected at the more northerly or more southerly array? Mm-hmm. And we used the release group as a surrogate of OMR. So we had those two release groups, and we compared the portions between those for every fish having a binary response of being detected more northern or southern array. We used it was a GLM conducted in R using a binary response and a binomial distribution. We checked for over-dispersion, and if it was over-dispersed, we use a quasi-binomial distribution to control for that.
So what's the sample size?
It depends on the junction. There's more at Turner Cut, and then there's fewer as you move along the San Joaquin River, and there's far less in Railroad Cut. We had about 74 or 75, I think, for Railroad Cut. In the South Delta where there was less fish, and we had larger sample size along the main stem. I think Turner Cut's more a couple hundred.
Yeah, and that analysis has to assume that all the fish are acting independently of one another. Do you think that's reasonable?
They were spread over, these detections usually occurred over a week. So some of there's probably some interdependence between some of the observations if they're moving through the junction at the same time. But there was a wide range of times where the fish moved through the junction. So for most of them, for a lot of the observations, it would be independent. But there would be some if they're moving at the same time, experiencing that same condition.
Thanks, David. Can you help me understand how you selected the release site and how that relates to where smolts are released, I guess, in the system?
I'll comment on what I can. I wasn't involved in that part of the process. It was designed in 2012 before I started being involved with this group. I think it was settled with the collaborative work team, which involved California Department of Water Resources, Bureau of Reclamation, NIMS, NOAA, and they chose that location. I'm imagining it's probably to fill in a void where there hasn't really been many studies before, and a lot of these steelhead are moving, some of these steelhead are moving along the Santa Joaquin River where they chose it. One of the project leads is here, so if you want, he could probably answer that question. It wasn't something I decided.
Yeah, I guess I was just wondering if that's a site where most hatchery fish are released, or just
no.
okay.
A lot of them are actually released down in Mossdale and up in the northern Sacramento. One of the reasons it might have been is we've been putting a lot more fish actually closer to the delta, so actually more will survive into that, because we really haven't had that much information on acoustically tagged fish in the southern delta because of mortality reaching there. I think that's probably the reason they did it. Thank you.
Thank you, David. I enjoyed your presentation. I have just a couple comments. A little bit of background on the first one. We've been doing the biophysical modeling that you talk about, and our conclusion is that fish have a repertoire of specific behaviors. You don't see all the behaviors until you get into complicated systems. And hydraulically, these are pretty simple systems, at least for the most part. So it probably wouldn't be a bad approximation to just take the cruising speed of a juvenile steelhead and add it to the particle tracking distances. It might give you a sort of a surrogate biophysical modeling approach. It would make that particle tracking data more useful. The second comment is the Corps of Engineers, as part of the Sac Bank project, is doing some of the same work. Now, of course, their system is linear and there are other differences. But if you want to pursue the biophysical modeling, it would probably be a good idea to coordinate with that ongoing studies so you can get the benefit of what they've learned.
Well, definitely. It's great to bring all the expertise, and it's going to take a lot of people working through to, one, identify what the behaviors are, what that full repertoire is, and which ones we need to incorporate in what systems. So I think it's going to take a lot of good people from various agencies to try to attempt this. I know Doug Jackson's going to talk a little bit about some of the starts that they're doing with this, and I think we all have to work together to try to solve this problem and make a couple of biophysical models.
Yeah, I was going to add to that. DSM-2 has kind of great, has a long history, but we have much better modeling tools available now. I mean, there are multi-dimensional, Michael McWilliams, three-dimensional untrim. I think it'd be really worthwhile to do that same thing in the context of the best modeling tools available.
I think it's a great idea.
Okay. Well, that's a good lead into Doug Jackson's talk. So, Doug, we'll let you take it away.